Welcome back, dear students. In this session, we will be talking about the different types of trapping methods of nematophagous fungi. Either they can capture nematodes by some structural adaptations, and sometimes they could be predatory, and sometimes they could also be endoparasitic to fungi, and they could trap the host and kill it. So the mechanisms of trapping, as we had told earlier, it could be adhesive hyphae, branches, nets, knobs, as well as rings, which could be either constricting and non-constricting. Now coming to adhesive hyphae, the nematode can come in contact with the fungal hyphae, and what happens, the worm remains in contact along with the hyphae because of a sticky secretion which has been or an addition which has been released by the hyphae, and this has been found to be yellow thick as well as a yellow thick chemical which will serve as just like a glue. And so the hyphal outgrowth, when it outgrows or when it's developed, what happens, more amount of these sticky secretion is being produced, and that would result into the complete trapping of the worm and inactivation of the worm. And sometimes you can also see the worm cell is being penetrated by the hyphae, and as the hyphae is penetrating into it, it will elongate as well as absorb the nutrients from the nematode, and that would ultimately lead into the death of the worm. So the production of the adhesive glue, or the adhesive, adhesive is a glue itself. So the production of this adhesive by the hyphae is mainly to keep the worm stationary or to trap it over there. Once it has been trapped, what happens, the worm is being inactivated, and the hyphae will penetrate into the worm and start absorbing the nutrients from the worm, and finally the death of the worm can be brought about. Now, this is a case of that where the nematode is getting trapped within the hyphae because of the adhesive produced by the hyphae. Another case is the adhesive branches. You can see that small and simple organ-like structures which will enable the capturing of the nematode has been possible. Now, these should be sometimes few cells in height, and they mainly arise from the main prostrate hyphae as short laterals or grow as erect branches on or below the substrate. And usually this is also being coated by a thin film of adhesive material, and some fungi such as the Dactyella are good examples which can produce that. Now you can have a look on this. That is, rather than the whole fungi to be involved in that's rather than the full hyphal element to form adhesives, some branches of it can produce some what adhesives, and that could help in the trapping of it. These have been found to be highly microscopic. That's what the image is depicting over here. So some branches of it will produce some adhesives, and that will in turn what, trap the nematode over here. That is the case of the adhesive branches, as we have noticed in Dactyella. Another feature is the presence of adhesive nets by certain fungi. Sometimes single loop-like or complex multi-branch networks are being formed, and attached to these networks there would be certain adhesives. Now, in the case of Anthrobotiris, you can see a similar adhesive net, and this action is similar to that of the adhesive hyphae, except that they never secrete adhesive material, but they get coated. Just have a look like this. They're not secreting it outside, but they just coat it here. Okay, that's the difference. So a net-like structure is being present here, and the nets are there, and within the nets the nematodes get captured. Now coming to the next one, the adhesive knobs. Just remember the knob-like structures or the door knobs. You can remember that. On and what are these additive knobs? They are usually distant adhesive globose or subglobose cells which have been produced by certain fungi, and their main action is the production of thin adhesive film over this knob surface, and so the nematodes will get trapped by the knob. And you can see that the nematodes can be attacked by several knobs, and the knob penetration and nematode death can be brought about. This is an example of a knob. Now you can see the knob like structure over here, and at this end, that is in the previous examples, you can see that either the whole hyphae will be adhesive, or sometimes a branch of it will be adhesive. Now here it is only the knob. This is the knob. The round structure is called the knob. Only the knob will be found to be adhesive, and the nematode gets attached over there. You can see it over here. Onto this knob, a nematode is getting attached. And it is not one single knob, multiple knobs are attacking it. And then the nematode is being killed. Now coming to the non-constricting rings. Sometimes you can see that erect lateral branches arise from prostrate, creeping the septate hyphae. And what happens, these slender branches widens and the branch curls to form a circular structure called the ring. The cell wall and the stalk and the tip of the branch fuses at the point of the contact, and three-celled rings with a stalk is being formed. And the nematode usually enters into this ring, and when the worm moves forward, what happens? It is marked constriction of the hyphal cuticle will be formed, and the nematode struggles. And what happens? It breaks up at the weak points of the ring, and the worm movement will be further tried through the ring. But what happens? The nematode will be finally entering into the ring, and finally the nematode death happens over here. So just have a look into this. These are the rings which we have here, and the fungal hyphae itself has been forming certain rings. Okay, and into that what happens? Certain nematodes can happen to enter, and they get constricted. Not constricted, that is when they move through that ring itself, the weak portions will be getting broken down. These are non-constricting rings. There are the other type of rings called the constricting rings. Now you go to look into the constricting rings. They are also being produced similar to that of the non-constricting ones. And here the supporting stalk is shorter and it has been found to be stouter. Now you just look into this. This is a supporting stalk. Okay, the supporting stalk of a constricting ring. Okay, in a constricting ring, it has been found to be shorter, and here it has been found to be stouter. This is the case of a constricting ring. Now, and usually they have been found to be three-cell rings, and Arthrobotrys are the species which mainly forms constricting rings. And the action of the ring is that once a nematode enters into the ring will swallow the nematode in a single hold, and the induction of the ring swelling is being carried out because of friction. And what happens? The deep constriction of the worm's body happens, and it struggles for a few minutes, but it gets inactivated, and the hyphal penetration and exploitation of the nematode happens. So in the case of a constricting ring which has been produced by a nematophagous fungi, you can see that a three-cell ring is being formed with a stalk. You can see it's a small stalk, and this is a nematode. When the nematode comes inside this see, inside this three cells will get constricted. That is, if a nematode has been present here, it gets constricted in between these three cells, and that it would kill the nematode into it will trap or constrict it. If you want to make more clear with it, you can have a look like this. Okay, and that would choke all and even kill the nematode. After that, see, these are all examples of constrict. The hyphae with the constricting ring which is trapping it, and it gets constricted and the nematode is being killed or inactivated. See, this is the entire nematode, and this is the hyphae with certain constricting rings which is choking and which is constricting once the nematode is being trapped, and ultimate death of the nematode will happen. Now coming to the endoparasitic fungi, some of the mycelium will be external to the nematode body, and they bring modifications in the conidia to kill their prey. For example, those fungi include cephalosporium, myria, etc. Uh, they produce some zoospores which will track down the worms by swarming and eventually encyst near the nematode's body orifice, penetrate, and they will colonize the prey. So they produce some spores which will track down the worms, and they will swarm and eventually what? Encyst the nematodes. These are the endoparasitic fungi which we have. These are some examples of it. The hyphae is penetrating into it and attacking it. Then we have the egg parasites. Some fungi, as I told you, they will be parasitic to the what? Nematode egg itself. Here the hyphae will develop a terminal swollen structure, and the hyphae is narrow, and it has a narrow infectious tube will be developed from the hyphae, and the tube will penetrate into the eggshell. The hyphae will swell up and form a penetration bulb, and from the bulb it develops numerous absorption hyphae, and finally the hyphae consumes the egg nutrients. You can have a look of Dactyla over here. If this is certain structures which will penetrate into it, into the what? Into the egg of the what? Of the nematode. The nematode's egg has been penetrated by the hyphae. I hope you got an understanding of what is nematophagous fungi and what are the different types of nematophagous fungi, as well as the various methods by which it can carry out the process of nematophagy. So with this we come to an end. Thank you.